Namaskar, welcome to CEC Gurukul Lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. Today our topic of lecture is going to be very interesting one. We are going to learn something very fundamental and something very basic whenever we want to work in a laboratory environment. Today we are going to discuss the basic laboratory instruments. When we will be covering this entire series of lectures on basic laboratory equipments, we will be able to understand the operation and maintenance of these basic laboratory instruments. We will also be able to perform various laboratory experiments which much more skilled manner. So what are these fundamental laboratory instruments? Basic laboratory instruments include the common instruments which we use in our day-to-day laboratory experiments, such as weighing balance. To work on any kind of compound or chemical, we need to measure how much amount we want to take. So this is one of the most basic instrument which anyone would use. pH meter is another basic instrument to measure the potential of hydrogen ion in any of the solutions. Magnetic stirrer, pipettes and micropipettes, autoclave, laminar flow hood, BOD incubator. The list goes on. The list continues with incubator shaker, micrometer, hemocytometer, spectrophotometer, agarose gel electrophoresis unit, SDS page unit, centrifuge, distillation unit, conductivity meter, lux meter, etc. So in this series of lectures, we will be discussing the basic principles of these instruments, the working principle also, the uses and limitations of these instruments. This will enable us to be able to handle these equipments in a much more skilled manner in our laboratory conditions. These knowledge of basic instruments is a must for any basic science student. So we shall start with the first and foremost equipment which any lab will have, and that is laboratory balances, also called as weighing balances. So laboratory balances or weighing balances, they are used to measure an object's mass to a very high degree of precision. They provide high readability, a broad weighing range and a high degree of accuracy. Weighing balances which are used in laboratory, they come with various specifications such as how much capacity of the weighing balance is, how much is the resolution, the width and length of the platform, etc. These are some of the pictures, diagrams of common laboratory weighing machines. Here on the left hand side, this is a top loading weighing balance and this is an analytical balance. Basically both of them are analytical balances only. So there are different types of laboratory balances which one can use in any lab or conduct any experiment, such as beam balances, equal arm balances, unequal arm balances, spring balances, analytical balances, moisture balances, top loading balances and platform balances. Out of various different types of balances, today we shall be discussing basically about analytical balances, how they function, and also we will be talking about top loading balances. So these are various different types of weighing balance which are used in a laboratory. This one is analytical balance, this one is equal arm balance or trip balance. They are also called as trip balance. This is a platform scale which are used for heavy mass weighing. This is a spring balance, top loading balance, triple beam balance and ultra micro balances used for, this ultra micro balance is used for very minute and very precise weighing of any substance. So what is an analytical balance? An analytical balance is a class of a balance that has a readability of 0.1 milligram or better than that. At this level of readability, it is required for the balance to have a draft shield. The samples are placed inside of the draft shield onto the weighing pan. So what is a draft shield? Let us understand this entire structure of a weighing balance with the help of this diagram. So a typical analytical balance will have this type of common structure, where this is a platform on which there will be certain display on which the weight will be displayed here. Then there is a tare button, power button, and then at the bottom there is something called as level adjustment feet. For a accurate measurement, it is very important that any analytical balance should be kept at a leveled position, and to maintain that level we use these screw, and this is the point where the level indicator is placed. Generally it has mercury ball inside, which indicates the level of this entire weighing balance. Now in the center there is a platform which is called as balance pan, and surrounding this balance pan there is box or chamber made up of transparent material, also called as draft shield. This transparent boundary made up of glass or plastic allows the material here not to be disturbed by any kind of movement of air. So this draft shield protects any movement of air, and by doing so the weight is measured at a much accuracy. So the purpose of the draft shield is to prevent drafts from influencing the weighing results. Most analytical balances are so accurate that even just minor breezes caused by someone walking by would disrupt the reading. This is why analytical balances have draft seals. Analytical balances are designed to measure small masses from around 320 grams to sub-milligram level. These are sensitive enough that they normally require a transparent enclosure, which we have talked about as draft shields, and these draft shields blocks air currents and prevent the dust collection. To perform well, these weighing balances, they have to be level, and so are fitted with level indicators. So let us try to understand how these analytical balances work. Balances do not directly measure mass. They measure the force or the weight that act downward on the balance pan. Most analytical balances are electromagnetic balances, and so measure this weight using an electromagnet. So let us try to understand this principle with the help of a diagram. So this is a schematic diagram of an analytical balance. So here the weight is put on a balance pan. So the force of this material, it will pull down, and there is an electromagnetic servo motor attached downwards. So this is the electromagnetic servo motor. Any difference created by the mass or the weight of the material will be detected by this electromagnetic servometer, and these electrical differences, they are converted into a display in terms of weight. In some of the weighing balances, there is a light source also, and there is a detector, so any change will cut out the light, and whenever there is a accurate measurement, the light will automatically glow. So the electric current required to generate this force is proportional to the weight, and so can be used with appropriate calibration to calculate the mass. This mass is then displayed on the screen. It also means that placing any magnetic materials or magnets near the balance could cause problems for the balance. One should wait at least 30 minutes for the electromagnetic field to stabilize before starting to work on an analytical balance. This time of 30 minutes may vary depending upon the manufacturer and the model of analytical balance which we are using. To signal when the weight and electromagnetic forces are equal, many balances have a null detector that uses a light source and detector. So by using this electromagnetic servometer, we detect the force applied by any matter over the balance span, and it is reflected in terms of digits on a display. So a variation of one such analytical balance is a top loading balance. So as they are often referred to as top loading, it is a balance that does not have a draft shield, and because there is no draft shield, the samples are loaded directly onto the weighing pan. These balances typically have a readability of 1 milligram or less, and it is a balance with an open weighing pan loaded directly from the top. So this was all about the basic idea about the laboratory pans, laboratory weighing balances, which we use in our day-to-day experimental conditions or experiments. Now the next basic instrument happens to be a pH meter. A pH meter is a scientific instrument that measures the hydrogen ion activity in water-based solutions. It indicates its acidity or alkalinity, and they are expressed as pH. The pH meter measures the difference in electrical potential between a pH electrode and a reference electrode, and thus the pH meter is sometimes also referred to as potentiometric pH meter. So this is the diagram. This is the picture of one of the commonly used pH meters. Here we can see that the electrode is dipped in the solution to be tested, and the pH reading will come on a display. Then one such field pH meter, this is one such field pH meter where the electrodes can directly be inserted into soil and the pH of the wet soil could be found out. So let us know more about the pH meter. The knowledge of pH is useful or critical in many situations, such as chemical laboratory analysis. The rate and outcome of chemical reactions taking place in water, they are often dependent upon the acidity of the water, and it is therefore useful to know the acidity of the water, or for that matter alkalinity of the water, and it is typically measured by means of a pH meter. So fundamentally a pH meter consists of voltmeter attached to a pH responsive electrode and a reference electrode. A reference electrode can also be called as unvarying electrode. The pH responsive electrode is usually made up of glass, and the reference electrode is usually a silver chloride electrode. Although in place of silver chloride electrode, sometimes mercurous chloride electrode can also be used. So let us try to understand the working principle of a pH meter with the help of a diagram. So a pH meter typically has two electrodes. The one electrode is called as the reference electrode. The reference electrode is dipped in a liquid medium or a gel medium, usually made up of potassium chloride, and they are generally made up of silver chloride. So this reference electrode, they do not have, or they are also called as unvarying electrode because their potential does not vary, they are mostly constant. Now there is a second electrode in any pH meter that is called as measuring electrode. Now this electrode is always subjected to the sample solution. At the end of this measuring electrode, there is a bulb-like structure also called as glass membrane. It has a similar electrode made up of silver or silver chloride, and the buffer of the solution present inside this bulb happens to be 7. So when this entire measuring electrode is placed in a sample solution, there is a change of voltage taking place, and this change in voltage is measured by a voltmeter. This voltmeter measures very minimal or minuscule amount of change in the voltage also, and this voltmeter gives us the reading of the change in the voltage, and it is displayed in terms of pH. So when two electrodes are immersed in a solution, they act as a battery. The glass electrode develops an electric potential, or we can say the charge, that is directly related to the hydrogen ion activity in the solution. To give you an idea, approximately 59.2 millivolts are created per pH unit at 25 degrees Celsius. So that was the basics about a pH meter. Another such basic instrument, and a very simple one, happens to be called as magnetic stirrer. Magnetic stirrer is one such instrument which is used to create or make homogenized solutions. The basic principle happens to be very simple, and the usage is also very frequent. A magnetic stirrer is a device widely used in laboratories and consists of a rotating magnet or a stationary electromagnet. These rotating magnet or stationary electromagnet, they create a rotating magnetic field. Under the influence of this rotating magnetic field, a small stirrer bit keeps on circulating in a rotating fashion, and it is immersed in the solution, and that solution also starts rotating in a vortex manner. This device is used to make a stir bar immersed in a liquid quickly spin or stirring or mixing a solution. So it is the stir bar which rotates under the influence of stationary electromagnet or rotating magnet, and under this rotational motion of stir bar, the solution also gets mixed up. These are the pictures of very commonly used magnetic stirrer. Sometimes the magnetic stirrer also comes attached with a hot plate, so we can warm the solution also, and at the same time here we can see that a vortex is created because of the rotational motion of liquid, and because of this rotational motion of the liquid, the dissolution of any solute takes place at a faster rate, and they are also homogenized very properly. And this magnetic stirrer, it has knobs to control the magnetic field and the speed of rotation of stir bar. There are certain magnetic stirrer which can have multiple platforms, so multiple samples can be mixed simultaneously at the same time. So most of the current magnetic stirrers, they rotate the magnets by means of an electric motor. This type of equipment is one of the simplest to prepare mixtures. Magnetic stirrers are silent and provide the possibility of stirring closely, closed systems without the need for isolation, as in the case with mechanical agitators. So that was magnetic stirrer. Now we shall move on to one of the most widely used equipment in any tissue culture lab. To transfer any material in a very sterilized condition, we use an equipment also called as laminar flow equipment or laminar airflow hood. Laminar airflow hood can be abbreviated as LAFH, and it is a primary control device which provides multiple services, such as it provides clean air to the critical sites, which are immediate aseptic compounding areas. It can also provide constant flow of air out of the work area to prevent entry of room air, and an outward flow of air from the hood that suspends and removes contaminants which have been introduced in the work area. So there are multiple uses of this instrument, which we call as laminar flow equipment. This is a picture of a laminar flow hood. So this is a cabinet under which there is a flow of air which is maintained, and this flow of air ensures that there is no contamination inside this entire chamber. So what are various uses of laminar flow? So it can be used where an individual clean air environment is required for smaller items. For example, particle sensitive electronic devices. It is used for special operations in laboratories also. It can be tailor-made to the specific requirements of laboratories and are also used for general lab work, especially in medical, pharmaceutical, electronic and industrial sectors. So a laminar airflow workstation is a closed cabinet fitted with HEPA filtered airflow system with unidirectional constant flow of air with almost no or minimal turbulence. The airflow velocity remains between 0.4 meter per second to 0.6 meter per second. So there is a constant flow of air, but the velocity is not very high, so that it could hinder in our working with any of the material under this hood. The purpose of using such workstations in laboratory is to create particle and bacteria free working environment to carry out specialized work. HEPA filter is a particulate filter which traps the airborne particles and microbes but allows the gases to pass through. HEPA filter should be fitted either at or near to the clean room inlet. A pre-filter is also fitted upstream of the HEPA filter, thus extending the final filter's life. A fan is also fitted which pumps the air through the filter. So we shall talk about this construction and the basic working principle of laminar flow hood in detail in our next lecture. We have just discussed in this lecture the basic instruments such as weighing balances, pH meter, magnetic stirrer, and the basic introduction about laminar flow. In our next lecture, we shall talk about laminar flow in detail and also about one of the very crucial instrument called as autoclave, which is used to sterilize any sample. So I hope you have liked this lecture. Thank you so much for listening.